Thank you so much. I want to begin by, first of all, thanking the organizers for giving me an opportunity to share my research with you. My area of physics is biological physics and computational biology, so this is a perfect venue to discuss this work. My lab is broadly interested in understanding design principles that underlie the assembly of biological structures. We are interested in intracellular structures or even tissue scale structures. Today, of course, I'm going to be focusing on intracellular structures. Now, our cells are able to create a range of structures within them. And in doing so, I'm going to argue that they're solving a variety of engineering problems. Um, I have classified the problem into three different categories here. From the same pool of building blocks, our cells are able to create structures that are same in size. We see that in flagella and chlamydomonas, which are the arms that they use to propel themselves forward in any kind of medium. These structures are made up of microtubules and they're always the same size. But if you move over to another microbe called Giardia, it makes four pairs of flagella, all different in sizes. If you look at stereocilia, it's a very interesting actin bundle, which displays a very beautiful staircase-like structure. If you see bundles towards the front of the structure, they're quite a bit smaller, but if you go towards the back, they're longer. So it's kind of neat that a cell is able to create structures like that. We also see cells make structures that have completely different geometries, but they share the same pool. An example is yeast the cables and patches in yeast cells. Now, each of these structures that I'm showing you right now has been a case study in my lab, and even though they appear to be from different model system, the question that we are asking from them is exactly the same. We're always trying to understand how are these different structures being able to get created inside of cells, but they're rapidly exchanging their building block proteins with their shared pool. In order to answer that question, our workflow has been the following. We first talk to our biologist friends and develop mathematical models that describe the effect of key molecular processes on the specific organelle. Then we validate our model with experimental data, and perhaps the most exciting part is to use our quantitative models to design new experiments with collaborators. Today, I'm going to be giving you one example where we were able to do all of that. This is Nucleolus. As you can imagine, this kind of work will require collaborations. So for this particular work, we partnered up with Dr. Stephanie Weber at McGill University. She's our experimental collaborator, and Yanni Kondev at Brandeis University. I also want to highlight two members of my team, Nishant and Rabia, who were star students who pretty much did all the work. So that was really cool to do this work with them. All right, let's talk about nucleolus. Our cells are full of all kinds of organelles, and nucleolus is one such organelle. It exists inside the nucleus. That's what you see in green over here. These are HeLa cells. The primary function of these organelles is to create ribosomes. So they're a big part of ribosome biogenesis. They assemble around our ribosomal DNA, which is inside of our nucleus. And the reason that we care about these organelles is that for the longest time, the size and their number were considered to be strong markers for disease like cancer. Turns out that they are very irregular and they are enlarged and they're more in number in cancerous cells. Of course, now we know that they're associated in neurodegenerative diseases as well and aging. So they're a super important organelle to understand the assembly of. For this work, we chose C. elegans as our model system, and it's an excellent model system for understanding a number of biological processes. And the reason being that it's transparent, so it's easy to image, and also like you can do a host of genetic manipulation on this particular type of worm. Its cell division process is also very interesting, as you can see over here. It starts its life as a single cell in a fixed size egg. But as time progresses, the cell kind of divides, everything gets smaller, and it gets smaller through reductive divisions. But in every cell division, essentially, this is what's happening. You start from a single, a single cell, P0. It divides into two different types of cell. It's an asymmetric division. So these cells have different volumes. But then AB type cells will divide even further and further, and that's how things will go on. And each of these different types of cell types will become a different part of the worm. we'll know in a while why this is interesting, but I want to especially highlight AB type cells, which will become hypodermis and the neurons, and the E cells will become intestine, for example. Now, let's look at what the nucleolus looks like in these systems. What we see is that in worm cells, especially, nucleoli don't really form properly until the 8 cell stage. And after the 8 cell stage, from the 8 to the 16 to 32 to 64, we can see that the embryo pretty much stays the same size, but the cells within them get smaller and smaller. So what you see over here in a circle is the nucleus associated with the 8 cell stage. And what is marked over here are these nucleolar proteins known as Fib1, and th those are going to be our marker for the nucleolar size. As the cell divides in this early development from 8 to 16 to 32 to 64, we see that the nucleus gets smaller and smaller, and so do the nucleolus amongst them. Now, you'll notice that there's always two nucleoli per cell, and that is because there are two rDNA sites where the nucleolus is assembling from. There's something else that we know about the system is that um, the Fib1 protein that you see inside of these embryos, this is all maternally loaded. So this is early development. So these cells have not started creating their own Fib1 proteins yet. They're still working off of whatever came from mom, basically. Um, and this particular paper that came out in 2015 made a very interesting observation. When they plotted the maximum nucleolar intensity, which is basically the sum of the Fib1 GFP that you see over here, um, that actually scales with the nuclear volume. Now, this is a very interesting observation, and this is exactly what you would expect if there was a limiting pool of nucleolar proteins that the nucleolus is drawing from. And in order to understand that, let's write a quick equation. I'm going to kind of go through the basics of limiting pole mechanism of organelle assembly. Let's assume that I have a structure that is composed of certain number of monomers, which can attach or detach from it. Now, the evolution of the size of that structure is going to depend on an assembly, and it's going to have a disassembly rate. But the assembly rate in a limiting pole mechanism is going to depend on the number of free monomers that are around. So that's this term over here. At steady state, if you assume a constant rate of disassembly, we can see that the steady state size of that structure is going to depend on the number of monomers that you have in the system, the volume, and the assembly and the disassembly parameter. Now, if I wanted to connect it to the nucleolus problem, I'm going to do a couple of substitution. I'm going to say that there's a constant concentration, so I can write N is equal to CV, and then I can write a dissociation constant, which is the ratio of your disassembly to your assembly rate. Now, if you do the substitution, indeed, what you're going to find is that your steady state size of your structure is going to scale as the volume of your volume within which the structure is assembling. To connect it to the nucleolus problem, you can think of the steady state size as the sum of the nucleolus within a nucleus, and this volume is now going to be the nuclear volume. So this is indeed what they saw in their experiment, right? So there was a straight line over here, and from the slope, you can actually find you can extract parameters like concentration and dissociation constant from the slope. So that's really cool. We were very excited about that. But limiting pool mechanism of organelle assembly also makes prediction about assembly kinetics. And it turns out that the nucleolus that we see in worm cells, they assemble much faster. So that was kind of a bit of a puzzle for us when we started working on this. We now know that obviously there are other factors contributing to this process, namely the process of transcription. So I wanted to begin by first showing you what nucleolar assembly even looks like in a particular cell. So these are AB type cells, and what you can see over here is that the nucleus is composed of some Fib1 proteins that's seen in gray over here. As time progresses, you see some passive assembly of little droplets, but after a point of time, two of those droplets become pretty big, and these are essentially what nucleoli are. They stay on for some time, and then after some time, they disassemble. With also fish experiments, people have seen that these nucleolar regions, like this nucleolus and this nucleolus, associated with where ribosomal RNA are. So these are, you can see in this experiment, there are a number of Fib1 type drops that develop, but the two that are associated with the ribosomal RNA are nucleoli. And this paper in PNAS in 2015, they did a really cool experiment. They actually stopped transcription from happening. And if you stop transcription through RNA interference, you saw, you can see in this plot over here that you see some passive assembly of little droplets, but no nucleoli at all. So that was very interesting to us. And, the problem the way that we looked at this problem was that we wanted to understand what role does RNA transcription play in the assembly of nucleolus. That's what we wanted to kind of figure out. And I just want to kind of give you a little visual of the assembly kinetics. So these ends are basically these little droplets that are growing over here, and nucleoli are the two big blobs that you see over here. And you can already see that there's a difference in the assembly of these two types of structures. This is assembling much faster than what you would expect from just simple assembly, simple limiting pool type assembly. So let's look at how the assembly looks like through development. And specifically, we'll focus on 8-cell to 16-cell stage just because we had more data for this stage. And what we're going to do is look at the assembly kinetics of different types of cells. So these cells that are marked in blue over here are AB type cells. They're more numerous in numbers. So every cell stage, every cell division, these 4 AB type cells will divide into 8, for example. And that's how cell division is going to proceed. So let's look at what nucleolar assembly looks like in different cell stages, right? So, there some 8-cell stage cells are marked in these colors, and AB, the 16-cell stages are marked in this particular color. Now, looking at this data, we can already see a couple of things. We can see that different cell types have different maximal sizes, right? Like all of these cell stages have different maximal nucleolar size associated with them. They have different lifetimes. And also, very curiously, the assembly kinetics has a specific shape associated with them. So this was very interesting to us, and we wanted to come up with a model where we can kind of understand where this actually could come from. So we came up with a model which was inspired from some of the some experimental observations that were already out there. We assumed that the growth of nucleolus is controlled by a limiting pool of one of its components. For us, it's going to be Fib1 proteins because that's what's marked over here, and that's what we had quantitative data for. And we also assume that the nucleolar assembly is nucleated by ribosomal RNA. There have been some papers, especially one in flies, which also suggested this particular mechanism, and we were curious to see how much this idea would contrast with what we see in experiments. Now, I want to kind of describe that RNA transcription proceeds in 2 steps. So the first step is the step of initiation. In that particular step, your RNA polymerase would land on your rDNA site. And as it moves along the rDNA site, you're going to start producing your nascent RNA transcripts. We assume that the nucleolar proteins or the nucleolus is going to ultimately condense and get nucleated on these nascent RNA transcripts. So that's kind of the basis of this model. So our research goal, just to state it again, is to develop models of nucleolar assembly that are driven by this particular process, initiation and elongation of nascent RNA. And then what we want to do is make theoretical models and compare our predictions with whatever experimental data that we have. So that's our main objective. So in the next few slides, I'm going to try to tell you a couple of equations that we wrote for droplets that are condensing. I'm summarizing years of work over here, but obviously the details are there in the paper, but I wanted to kind of tell you what the key points of the model are. Now we know that droplets or nucleolar droplets, they form even without transcription. So we assumed an equation for the droplets that proceeded like this. It's a very limiting pool type of model where you have an assembly rate that depends on the number of free nucleolar proteins that you might have. And we assume the disassembly rate to be constant. With transcription, you're producing these RNA transcripts, right? And we decided that nucleolus is going to be the collection of droplets that nucleate on this particular RNA, right? So each of these droplets can grow and decay independently, but the number of those droplets is going to depend on the number of RNA transcripts that you have. So that's the key idea of this particular model. If you put all these things together, this is the basic equation of nucleolus that we're going to work with. So this describes how the nucleolar size changes as a function of time. And it has two components. One component is that it's going to depend on how many free Fib1 proteins that are there. And it's also going to depend on how many droplets are contributing to the nucleolus at that particular time. So it's a combination of both those factors. All right. So now we have an equation, a working equation for our nucleolus. Um, we can kind of consider two different ways in which nucleolar assembly can proceed. One situation would be where the nascent RNA has completely reached a steady state, like the transcription is completely finished. And all that you have is different nucleolar droplets kind of condensing on that RNA template. And another situation is when the transcription is happening, the process of transcription is happening, and your nucleolus is also assembling at the same time. So just to put it in perspective, I have a couple of pictures to kind of show you what I mean. In one case, we are saying that the RNA transcripts are already there. We have the RNA template, and all that we have is little droplets kind of condensing independently on that template. The second situation is when the template itself is growing with time, and because of that, the number of droplets is also changing with time. So in one case, your number of droplets is going to be constant, but they're going to be growing independently. But in the other case, the number of droplets is going to actually depend on the process of transcription. So just to kind of reiterate, there are two processes that contribute to transcription. So you have a process of initiation, and then you have a process of elongation, which are denoted by alpha I and alpha E over here. We are assuming that both of these processes proceed as a linear function of time. So the rate at which RNA polymerase kind of land on the polymerase, it's a linear function of time, and the rate at which the RNA polymerase is moving along, that is also a linear function of time. We have some reasons for assuming this. There were some experiments done on flies where they actually measured the loading rate of RNAPs, and indeed they saw that there was a linear rise in that particular rate. So it's kind of based on that observation. Now, our number of droplets is going to depend on the amount of RNA around. So I hope you can appreciate that because of these two processes being a linear function of time, the amount of RNA and the amount of the number of droplets associated with them is going to grow as a quadratic function of time. So in two of these models, if you put the ingredients together, we come up with two different ways in which nucleolus kind of can evolve, right? And both of these make two different predictions for what the nucleolar size would be. I hope you can notice that there are a number of parameters that these models share, like the number of nucleolar proteins, the dissociation constant, volume, that's same in both the models. But the main difference in both these models is the assembly rate, Kp and Ka, and in the way that it proceeds as a function of time. In one case, you proceed as a negative exponential of time, but the other one, you're proceeding as a e raised to the power of minus t cubed. So it has a different time dependence. And these two are basically prediction for what the nucleolar size might look like. So what we did next was to compare that with experimental data. And in order to do that, we made use of a couple of estimations that we already had. So nuclear volume is something that we could measure directly from experiments. So we basically had nuclear volumes for all different types of cells at different cell stages. From published data from a previous paper, we could estimate the number of cells in one type of cell. And by assuming that all of these cells are at constant concentration and using their respective volumes, we could have an estimate of how many nucleolar proteins existed in every cell stage. We assume that the KD is the same, and we had KD associated with one of the cells from our previous paper. So we basically had most of the parameters kind of estimated. The only thing that we were not sure of was the assembly parameter, which is the Ka and Kp. So in order to extract that, we went to a particular cell stage, and that's what I'm going to show you right now. This is the E16 cell stage. And we compared the prediction of both of the theoretical models with the experimental data, and we noted a couple of interesting observations. While both of the model predicted the same nucleolar size, their dynamic behavior was very different. And we saw that active model, in particular, the model which assumed that your template was also growing with time and as were the number of droplets condensing on it, that was able to correlate with the data much better than the passive assembly model. So that was really interesting to us. And by extracting this particular assembly parameter, we wanted to see if the same assembly parameter, how would that compare with the assembly with other cell types. So this is E16, but now we're going to see data that comes from AB8, AB16, and all of the different cell stages that we have over here. So that's what I'm going to show you in the next slide. So let's walk through it kind of slowly. So this is your cells going from 8 cell stage to 16 cell stage. And each of this data point that you're seeing over here comes from a specific cell stage as is written in the title. In gray are the individual trajectories. And in the blue that you see over here is the average of all of these different nucleolar sizes. What we saw, which was very interesting to us, was that even though the cell types have different maximal sizes, all of these have different maximal sizes, but their essential growth rate was the same. The same Ka that we had extracted from E16 type data worked for these cell stages as well. And another curious observation that we made was that nucleoli seem to disassemble around the same time as the steady state is predicted by the model. So that was kind of a curious observation to us. In fact, if you take the equation and analyze them in early times, both the models make a different prediction for the growth scaling of the nucleolar sizes. And if you take the data and you arrange them in a specific way, where you take the nucleolar size, divide that by the steady state size predicted by the model, and then multiply that by the respective volumes, you will see that all of the data from different cell stages kind of collapse in initial times. And that was really interesting for us to see. So in dotted line, you see the prediction that you would expect for the passive model, which assumes that transcription has already reached a steady state and you just have independent droplets are condensing on top of it. But active model is where your template is increasing with time, and so is the number of droplets as well. So it seems like the active model does a better job of explaining the early assembly of all of these lineages. So we thought that was quite interesting. Now, another perturbation that we tried was to compare our predictions in situations where the embryo sizes were different. So we used this method of RNA perturbation, and we were able to create embryos that were two little bit smaller in size and an embryo that was bigger. And using the same mathematical model and assuming that only volume is changed, we predicted a couple of things. We predicted that there would be faster nucleolar assembly, and the size of the nucleus is going to be larger in smaller cells. So that's what you see over here. This is your control. These are the ones that are smaller than control, and they are showing larger sizes. And we also predicted that the size is going to be smaller in an embryo that is bigger in size. Now, we were able to see the same qualitative difference in the data as well, which was very encouraging. But when we compared the data to individual conditions, we saw some interesting observation. While while in two of the conditions, the scaling was similar to what you would see in the predicted scaling was similar to what you see in experiments. In one of the condition, this is the condition where the embryo size is the smallest. Nucleolus seems to be assembling even faster than what we predict. We have a couple of ideas and we tried them, but this is still a big mystery to us as to why this is happening. So yeah, this was very interesting. But again, if you take all of the data and you arrange them in the way that you take the size ratio and multiply that with the nuclear volume, we do see that there is two of the conditions match with what you would expect for the active model. So I hope I gave you a sense of what this project was all about. So I'm gonna quickly summarize. Um, this personally for me, I think this was a really cool problem because I really like the fact that this is an example where active processes like transcription play a role in the assembly of a structure. And I find it personally very fascinating that transcription mediated nucleation can be a knob to not just control where assembly of the structure happens, but when. So the placement and timing both can be controlled by a particular process. And I thought that was really interesting. I also find it very curious that in all of these cell types, the nucleoli seem to dissolve shortly after the predicted steady state size is reached. So we are currently trying to understand mechanisms that are affecting the nucleolar disassembly and the nucleolar lifetime. So this is still a work in progress for us. But I find it, personally for me, I think the fact that RNA transcription can play a role in controlling the timing and placement of assembly of a particular structure is really, really cool. So with that, I'd like to thank my collaborators again and my organizers for inviting me to talk, and happy to take any questions.
Thank you, Lishi, for a wonderful talk. So we have some questions from the audience. I'm going to ask them right away. Okay, so the first question was from Jing, and she was asking, why does the disassembly rate not scale with M?
Yes, I think that's a very good point. In this version of the model, we kind of assume it to be constant, but this is something that we are actively looking at right now. Like if the disassembly rates of these droplets, dependent on the size, that would predict a different scaling behavior. So we are interested in kind of checking that with further data.
Thank you. Then the next question was from Miranda, and she was asking why nucleation on nascent as opposed to mature RNA transcripts. So, why is nucleation happening on nascent RNA transcripts? Is it because the RNA decays or becomes a our RNA decays or becomes a ribosome too quickly once formed, or is it length driven?
That's a very good question. So our assumption was kind of guided by the fish experiments that they did. So in the fish experiments, the nascent RNA co-localized with nucleolar assembly. So that's why we assumed it to be the case. And it turns out that this assumption is, it's matching well enough with the experimental data. So it feels like that's a reasonable way to proceed.
Thank you. And then Guangxi is asking, could the faster than expected growth you showed in the end be explained by phase separation?
Oh yes. Oh, I would love to know. So I'm open to any ideas over here. This is a mystery to me right now. One idea that we are thinking is that the IMA is an import factor. So IMA3 is an import factor, which kind of tells you things that can be imported inside the nucleus. So when you're knocking it down, we believe that it might be affecting transcription in some way that affects the assembly of nucleolus. So I'm not really sure what's happening in that situation, but it is a big mystery for us.
All right, and we'll take a final question. This is from Saptorshi, and Saptorshi is asking, I'm wondering about the identifiability in the parameter space. In both the active and passive models, what are the dimensionalities of parameter space? Do the model predictions uniquely match with the experimental data? And then following up on that, how does noise come into the picture? Is it
Yeah.
inconsequential for this particular case during assembly?
Yeah, those are very good questions. So, I just want to tell you that in this particular model, the, like the volumes are already measured, right? So volumes are already measured. The number of proteins we estimated from different ways. So there are no fitting parameters in this model at all. So that I find very, very interesting in this particular work. There is, when we did the estimation, we find like there are some bands associated with every estimate, and we've played around with, the uncertainties, and it seems like at least for the average data, we do a pretty well job of kind of correlating it. The second question about the variability is a very interesting one because we do see there's a lot of variability in nucleolar sizes, and that's something that I'm actively thinking about right now is that does this variability, is it just due to population level noise, or is it because of the stochasticity in the transcription process that gives you such a wide range in nucleolar size? So that's something that we are thinking about right now.
Thank you, Lishi. Thanks again, Lishi, for a wonderful talk, and audience for your great questions. Let's thank